Hello, and welcome to this webinar titled Raw Material Lot Variability Challenges During Manufacturing of Biosimilars Mab, hosted by Biopharma Asia Magazine and presented by Sekhar Reddy, DSP Lead for Biosimilars at Biocon MSAT. My name is Stephen Edwards, and I'll be your moderator. Now, please allow me to introduce our presenter. Sekhar Reddy is the Downstream Lead of Manufacturing Sciences and Technology for Biosimilars at Biocon Limited. He has over 10 years of experience in research and development, manufacturing plant setup, clinical and commercial manufacturing, and MSAT departments. In his current role, he leads the MSAT downstream mammalian team to support commercial manufacturing towards root cause investigations, process and cleaning validations, risk assessments, extractable and leachable studies, and alternate vendor qualifications, etc. Sekhar has extensive experience in laboratory and commercial scale equipment qualifications, MSAT lab setup, GMP, troubleshooting of manufacturing operations, and support regulatory filings on CMC sections. Sekhar Reddy has completed his Masters in Biotechnology from JNTU, Jawaharlal Nehru Technological Institute, Hyderabad, India. I will now be handing over to our presenter. Welcome, Sekhar.
Thank you, Stephen, for introducing me. Hello all, this is Sekhar. I am working in Biocon MSAT. I lead downstream team working for monoclonal antibodies. And today we are going to discuss about raw material and consumable variability during manufacturing process of monoclonal antibody. I am just moving to the slide number 2. Basically, so this slide talks about the Biocon as a company is working from India. So this is started in 1978 by Kiran Majumdar Shah. So we have over 10,000 employees, and this has global scale manufacturing facilities. And recently we have approved 3 molecules across Europe and US FDA regulators. One is Downstream biosimilar and PEG-GCS biosimilar. And we have insulin glargine as well across regulatory markets. So our company has nominated as the number 7th in the Science magazine in 2008. So moving to the slide number 4. So we have a state-of-the-art facility for working for insulin as well as monoclonal antibodies. Our operations are from India as well as Malaysia. We have the biggest insulin facility available in Malaysia, and we have large scales coming up for the biologic facilities in India. And this is to just give the background of Biocon, what Biocon is doing. So moving to the raw material challenges. So today's presentation, I am going to give a case study for 3 case studies we come across in the manufacturing of one of the Mab. So the question is, why would we need to worry about raw material variability, though they are meeting the acceptance criteria and released for commercial use? So we are going to discuss about 3 topics, how they are going to have impact on the routine manufacturing process. One is so quality compliance issues. As you most of you aware that the quality compliance issues, when you see an issue with the raw material or consumable variability, we need to raise a complaint to the vendor, and we have to have multiple follow-ups with the vendor towards resolving the issue. And we have a lot of documentation burden as well in resolving those issues. And we have next one, process inconsistency. As you know, due to inconsistent input quality material and consumables, the outputs of these steps would vary. So eventually we may end up in out of range scenarios. So this is coming under process inconsistency. And we have lower productivity. Some cases we may lose the product. So one of our case studies talks about how raw material or consumable variability would impact on the productivity. So we'll see one of these cases. And coming to the next challenge, impact on the critical quality attributes. As you know, this has huge implication on the final safety and efficacy of the product. Therefore, keeping the CQAs in control is very important in a biological manufacturing process. So sometimes even consumable variability or raw material variability have potential impact on critical quality attributes as well. And we have operational challenges. So today, one of the raw material variability, how this is having a operational challenge in the manufacturing setup. We are going to discuss operational challenge as well in today's presentation. And we have also two more challenges we face because of this: resource and root cause investigations. When you have multiple issues with raw materials, you have to spend a lot of resources, people resources and technical resources towards the root cause investigation and finding out the root cause. So as you most of be aware in the biological industry, finding out the root cause is not as easy. You have to go into deep of the process, understand where the variability or where the deviation is coming from. And attributing to the variability of the raw material is not an easy job until you do complete thorough investigation using different risk assessment tools. Sometimes it can take months to get a root cause if it is a lot variability, because we use hundreds of raw materials in the process, and attributing to single raw material is not easy job. So you need to have adequate resources for the variability situations. And the final one, process changes and regulatory impact. Certain changes, we probably cannot correct it. So in that sense, we may need to update the regulatory agencies stating that there is no choice, we have to adjust the process to meet our targets. So we may have pain in the regulatory questions, regulatory complaints issues as well. So these are the potential challenges which could anticipate if we have variability coming from the raw materials and consumables. I just have highlighted what are the typical challenges people face. So in this presentation, we are going to discuss about 3 topics. Topic number 1, variability in depth filter lot. So depth filter, I mean for removal of cell debris from the monoclonal antibody process. So topic number 2, we are going to discuss about the variability coming from chromatography resin lot. So basically this talks about ion exchange for purification process. Topic number 3, we are going to discuss one of the critical buffer components used in the elution buffer of one of the chromatography steps. So we will see each case how this has an impact on the manufacturing process on routine basis. So we will move on to the case 1, which is depth filter lot. Right. So in the monoclonal antibodies, people typically use either centrifugation followed by depth filter or primary depth filter followed by secondary depth filter. So in this case, for this particular map, we use centrifugation followed by depth filter, which acts as a secondary depth filter, which rates from 0.1 to 1 microns. And if you see the particular graph, wherein you can see there are 4 batches where the pressure levels are too high. So what we have observed is we have seen clogging. So this observation was after 30 or 40 manufacturing batches we have taken from the commercial scale. So it's a 5 consecutive batches wherein we have seen the clogging. So what we have done, immediate action, so we have reduced the flow rate, one of the steps. Then we increase the venting frequency to avoid any air bubbles have gone inside the filter. And we reduce the feed flow rate as well. And none of the action plan has helped in correcting this issue. What we have done, we have taken the further investigation to determine the root cause, what would go wrong in this specific case. So we have looked at multiple aspects which could potentially impact the clogging. So first thing coming into mind is that is there any variability from the process, basically feed variability. So you see in the cell culture process, you see cell count, viability, turbidity and other impurities are coming from the process. We have verified complete cell culture process by doing multivariate analysis whether there is any specific issue is leading to or is resulting to this clog. And we also seen filter connections between multiple parallel connections because this is almost 2.5 kL process. We use multiple depth filters. If we have seen there is any change in the way the procedure is made between the connections. Basically silicon tubing or braided hoses between the filter. And verification of operational product parameters. Have you increased the flow rate? Have you reduced the surface area? Is there any change in the surface area being used in the routine manufacturing process? And finally, we have looked at the depth filter loss variability. So these are the areas we look for everything. Finally, based on the review, we have identified that the probable and potential root cause for pressure is basically changing the lot, specific depth filter lot. So next slide will talk about how the depth filter lot has been identified as a root cause in this case. So look at the graph. So these graphs basically explaining the clearance of FCDNA. So I think some of you are aware that depth filters typically removes the host cell DNA because depth filters are charged filters. It binds the DNA. So what we have observed in this, we have looked at multiple lots previously, and what we identified that this specific lot, which has been used for almost 4 or 5 batches, has seen significant clearance of FCDNA. So the filter rate coming from the depth filter has very low level of FCDNA. And we didn't understand why the FCDNA levels are so low when compared to historical batches. Then we realized that the filter, the feed FCDNA is still same. The DNA coming from the cell culture process remains same. The variability that we have observed is post-depth filter, wherein the DNA levels are too low when compared to other batches. So what we identified is that this specific lot, the clearance is very high. Therefore, we went and checked with the filter supplier, and this looks everything is normal. You know that depth filters are nominal filter, it is not absolute filter, and you expect so much variability from the filter manufacturing as well. What it does, it is doing the specific charge which has been added to this specific lot probably a little high, or maybe the charge itself probably is different on the filter. What we identified at the end, the clearance is more, which is causing very high viscous environment inside the depth filter, leading to the high pressure in this specific case. The moment we changed the specific lot and whatever leftover filters we have not used, the moment we change the filters and the process come into normal. But keeping this variability in mind, what we have done, we have improved additional 30% safety on the existing safety. So basically, initially, while developing, we already made 50% safety on the worst case. On top of it, we have included additional 30% safety. Overall, so we have 1.8 safety factor. So far, we have been manufacturing more than 150 batches after that issue. We are not seeing any issue in the depth filter. So what it means is that you need to have significant amount of safety in case of depth filters. So because in this case the product cost is higher than the filter cost, and we could accommodate, or we could take this call of having higher surface area. So we don't see any product impact on the product quality. So we see only the yield loss because some of the product has left in the filter. We could not retrieve that product from filter after choking. So this is one of the case study what we have seen. Coming to the chromatographic resin. So purification process, you know that we typically use basically downstream works in 2 ways. One is filtration, and second one is chromatography. Both will play a very significant role in removing the impurities. One we talked about filter, so other one is chromatographic resin. So if you see the specific graph, it's very evident. All the dotted black lines indicates one lot, and remaining all other colors indicates one lot. What it means is that when you have different phases of chromatographic steps, the pressure across batches with the specific lot has shown very high. And towards the cleaning phase, the phase 7 and phase 8, the pressure is going very, very high, for which the chromatographic skid is unable to support to deliver the specified flow rate. Basically what it means is the flow rate is coming down because the pressure has gone more than 2.5 bar. What we have done, so we have investigated to understand the cause for this higher pressure, and we have taken the old lot and new lot, a specific resin, and we have done the pressure versus flow curves at small scale. In all incidents, it clearly indicates that the pressure from the new lot is significantly higher than the old lot. So we further investigated this vendor, what does it mean? So we went back and seen the resin certificate of analysis. what it means, why there is a difference. When we looked at the certificate of analysis, all the tests specified by the supplier are meeting and matching. Therefore, there is no specific issue has been looked at in the specific COA. But the difference we would have seen is that if you see the specific lot 2 has there is some flow rate limit for the resin, that is 400 to 700 centimeters per hour at 1 bar pressure. So we have to get that much flow rate when you set the flow at 1 bar. So old lot used to give 540 centimeters per hour. When it comes to new lot, only 462 centimeters per hour has come. It means that 540 and 462 both are passing, but 462 on the lower side. It indicates that this specific lot has the particles lower than the average. The overall particle distribution is different. Although this is 45 to 165 micron, the number of particles which has lower size are more. This is the conclusion we made, and there is no choice because we can't afford to discard the resin. So what we have done, we have modified the process flow rates to accommodate that flow, to accommodate pressure. Basically, we reduce the flow rate by keeping the residence time constant, and we try to adjust the process considering some safety. So we have taken the safety into account for 400 centimeters per hour. So if the resin in future, if resin supplies with 400 centimeters per hour, even that flow rate has to be supported with this flow rate. So we have considered a worst case as 400 centimeters per hour. We designed the flow rate, and moving forward, we have corrected the flow rate and continuing, and therefore we are not seeing any issues in future. So what we suggested here was, so whenever you get the resin, you just perform a scale down run and establish standard pressure versus flow curve and identify the specific new lot has within that pressure flow curve. So if you don't get in there, either you have to adjust the process or you have to tell suppliers, so we can't use this specific lot. There is only one choice. So only one important thing is during process development itself, we recommend to test at least 3-4 lots and see lot to lot variability with respect to pressure coming from the process. So this is the case number 2 that we have seen in our manufacturing process coming from the chromatography resin. Coming to the third case study, which is this is specific to one of the raw material, that is citrate component. Then we use this in the elution buffer of one of the chromatography steps. If you see the chromatography columns, so you can identify these are very large scale columns. You see very brown color inside the resin. So basically resin is completely transparent and whitish in nature, but the resin is completely turned down into brown. What would be the reason for this? Why the column has turned into this color? See, so what happened, one of the batch, when we passed on to the product load and buffers, so operator had identified that resin color has been changed completely. Then it has come to the MSAT team for the further investigation what has happened in this batch. So we started investigation from all the aspects of potential causes which could lead to this issue. So we have seen equipment, hardware. So typically what happens when you have so much excess equipment, even rusting can cause this kind of issue. So in that perspective, we have seen column hardware, process equipment and utilities, basically water plant and other stuff where the specific any rusting is there. We have verified all the equipment, we opened up the equipment, we verified physically and visually we don't see any difference in the rusting part. So we also check any unique contaminant coming from the process intermediates. So we also check the load, whether load has any different color. We have passed that load into UV different wavelengths and see whether any color absorption is there. And we also see process consumables, anything coming from the bags or the filters or tubings, etc. And finally, we have collected all the process buffers and solutions which were used for the specific batch. So coming to the conclusion, what we have done. So what happened? So each component, everything has been ruled out. So we have ruled out process equipment, we ruled out process intermediates, we ruled out complete hardware and process consumables and utility parts. and we come to a stage wherein we have to do some studies for the buffer. This specific buffer has 5 individual components being used to make a buffer. So what happens, each component we have identified separately, each component we collected from the warehouse as the specific lot and has been passed on to the chromatography column. If you see the right side, this is a scaled down column which was done at R&D scale. It's shown the brown color again. So what we identified, out of 5, one of the component has, when we pass one of the component into this specific resin, it has turned into complete brownish color. At the same time, when we filtered that specific component using the 0.22 membrane, the membrane has turned in completely brown color, which indicates there is some issue in the specific salt being used in the specific buffer. So, what does it mean? Where it is coming from? So, what we have when we investigated along with the suppliers, they indicated that they have done investigation from their side. So what we have seen, so they identified some ferric citrate. Ferric component is very high in the specific lot. So they also identified that there is some change in the extraction step in the manufacturing of citrate. So when we tested the buffer, so we have seen that iron content is marginally higher and the nickel, chromium also higher, indicating rust-like material in the specific lot. So we have come to a conclusion that this specific lot has some issue during the manufacturing process from the supplier. So when we visually verified this lot from one good lot and one issue lot, if you see the right side, you can see the pictures of 2 different lots. One is amorphous, one is crystal. What we used to see in the routine batches, in the normal lots, the salt used to be amorphous like powder-like. This specific case, what we have seen is completely crystal. So, but there is no testing in QC to differentiate whether this specific salt has to be powder or crystalline. So, they don't have any segregation. Therefore, this lot has been used and released into the manufacturing process. So, what we have done, we have communicated to supplier and as well as internal QC to include a visual appearance test in the COA. Therefore, moving forward, whatever lot comes to us, it has to be amorphous. It cannot be crystalline. So crystalline means there is some issue in the extraction step of the manufacturing process. So coming to the impact part. So what does it mean? So this is a issue with the purification process. So fortunately, we have a TFF step after the chromatography step, wherein whatever the ferric citrate or you have individual ferrous or citric ions has been filtered, buffer exchanged and removed. Finally, we have tested the product by IC2OES for the iron content, chromium and nickel content in the product and tested along with the controls. There we don't see any problem in the final product. So everything has been cleared in the TFF step. Potentially this has huge risk on the product because this is a product contact material. So overall conclusion, what I would like to communicate to you guys is, so controlling the raw material consumable is very critical in the process because it has huge ramification and implications in the manufacturing process. You need to have a lot of resources, you need to have a lot of money to investigate to issues, and what I recommend is when you do this kind of, when you do this biological process, ensure when selecting the vendor, ensure proper vendor where you get quality product by doing multiple vendor selection procedures, and you have to have adequate testing procedures to make sure that you don't get unacceptable variability between lots and batch to batch. So coming to the acknowledgement part, I would like to thank Kala Chandramouli. So she's head of MSAT Biocon, and Sagar Jawar, he's head of production, and Saurabh Patil is my colleague, and Vijay MS is head of production. All have played very important role in investigating these cases and show a permanent solution against one of these issues. and I would like to acknowledge all of these guys. And I also empathize that what we are working with the specific MAB, we are working with Mylan. They are partners to Biocon, and they are going to market this Biocon products in the US and Europe and other regulated markets. So I'll hand over to Stephen for the question and answer session.
Thank you, Sekhar. As you mentioned, now we will begin the question and answer segment of the webinar. And once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question. Why would raw material variability be a concern for a drug manufacturer? That question again for you, Sekhar, was why would raw material variability be a concern for a drug manufacturer? Once more for you, Sekhar. Dr. Reddy? The question is...
Yeah, so can I answer?
Yes, please. So why would raw material variability be a concern for a drug manufacturer?
Yeah. So as I explained earlier, this has very significant impact of a drug manufacturer, because as a drug manufacturer, you have to make sure that you get low variability from consumers or raw material suppliers to ensure your business continuity. So you should not have any challenges while patients need a essential drug, and therefore if you get so much variability in the process, you will not be supplied to the product because you have a problem in the manufacturing process. You have to stop the manufacturing. Therefore, it's very important to control the variability to the acceptance levels. We can't control the variability to negligible. You can control the variability to the acceptance level. From the business perspective and resource perspective, from considering the importance of the essential drug for the patient.
Thank you. Our next question is, what kind of measures should one consider in order to control RM or consumable variability?
So can I repeat, Stephen?
So that was, what kind of measures should one consider in order to control RM or consumable variability? Yeah,
basically, so you need to have robust vendor selection procedures in place in the quality system. And also you can do a holistic risk assessment to be carried out towards the segregation of critical and non-critical raw materials, keeping your process in mind. And when you have critical materials, you need to have detection control measures before it comes to the manufacturing release. And you need to also have the continued process verification of your process. Therefore, you will identify the out of trends, and you can have thorough investigation to attribute whether it's coming from any particular raw materials or consumables. So you need to have multiple things in place to control this variability for the routine commercial manufacturing.
Thank you for that answer. Our next question is, how much safety factors is normally considered for a depth filtration area during harvest clarification?
Yeah, there is any rule to state this much safety is required. Typically, what I have seen from different suppliers, I have seen suppliers where 20% safety is enough. I have seen some suppliers say 50% safety is enough, because this particular number has to come from the process development, process knowledge. So in our case, so we have given 50% safety factor when we develop. So what happened? We don't see any issues after 30 batches. The 50% safety is very well enough. After 30 batches, one specific lot has created so much problem in the manufacturing. Therefore, we ended up in having 80% safety. right? So it's good. So we have 80% safety. Then after that, we have taken 150 batches, and we don't see any issue coming from this issue. So you need to have permanent solution by having good safety. Keeping, whereas, for example, your facility also has to fit for the adequate safety. Or else you have to see alternate ways of purifying this. You need to have introduce centrifuge, or you need to reduce the flow rate, or you need to improve the feed quality. It's purely depending on the nature of the products and business perspective and cost aspects. There is no hard and thumb rule you have to keep 50% or 80%. So we need to work it out case-to-case basis.
Thank you. And that will conclude the question and answer session. But before I finish the webinar, I would like to ask our presenter, Sekhar Reddy, if you have any closing remarks.
Yeah, so I explained you earlier, so biological manufacturing process is very key and a very expensive process, and the drugs are very, very, very high costly in the market. So keeping raw material variability minimum is one of the very important aspects of the business continuity. I suggest viewers is that you make a proper and holistic risk assessment for all the raw materials starting from upstream to formulation stage, and identify the critical raw material and try to have alternate raw material where you feel that some raw materials are critical, some consumables critical. Whenever you see a variability in one particular vendor, you can move it to other vendor. That is one of the important aspects you have to keep in mind. Every critical raw material has to have an alternate vendor. And second point, so you need to have robust communication process between suppliers and quality people. We need to know what is happening in the manufacturing process. We need to have frequent interactions with respect to vendor audits, site audits, and we need to have communications, what overs are happening at supplier side and what deviations are happening at supplier side. We have to keep engaged with the suppliers in order to have the consistency. And also in process, also we need to have robust testing procedures based on the criticality of the raw material to ensure the failed material will not come into the routine manufacturing use. And keep key documentation practices and robust risk assessment practices, and keep update and work on the alternate raw materials and impact on the products always.
Well, thank you for that. And I would like to take this opportunity to thank our presenters once more, that's Sekhar Reddy, for sharing his knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions for our presenter over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers for you. Thank you, everyone. Have a great day.